I just realized I cut out. I'm gonna change my Wi-Fi. Give me a minute.
Okay. All right, so we'll get started in just a couple minutes. We'll give people 1 or 2 more minutes to join. Ooh, already a question. Wow. All right, we'll get started in just a minute. All right, perhaps in the interest of time, we go ahead and get started. Welcome. My name is Katie Geringer. Thanks so much for joining us tonight for a journey into pooled COVID saliva testing to see what happens to your saliva sample each week. This virtual lab tour is sponsored by the Wellesley Education Foundation, which has provided funding for innovative program in the Wellesley Public Schools for over 35 years. In the last 10 years, WEF has funded nearly $3 million in educator and district-wide grants, impacting every student in the Wellesley Public School system. This year alone, thanks to the generosity of our community, WEF has contributed $600,000 in support of Wellesley's viral testing program, and will be providing nearly $80,000 in additional funding this spring in support of educator grants. WEF's See Yourself in STEM programming during the month of April is aimed at encouraging and supporting youth from all backgrounds to pursue STEM careers. Despite representing an increased portion of the overall workforce, women, people of color, first-generation students, low-income individuals, English language learners, and people with disabilities remain largely underrepresented in STEM fields. In partnership with Dr. Charmi Curie, Wellesley Public Schools Director of Diversity, Equity, and Inclusion, the WEF See Yourself in STEM program will help fund impactful initiatives that further the mission of helping more people see themselves in STEM. Those who wish to support this work are encouraged to make a donation by visiting WEF's website at www.wellesley-education-foundation.org. Tonight, we're very excited to have Dr. Prem Premsrirut here with us tonight. Dr. Premsrirut is a co-founder and president and CEO of Mirimus Incorporated, a company at the forefront of developing RNA interference and CRISPR gene editing technologies and investigating their potential therapeutic applications. At the start of COVID-19 pandemic, Dr. Premsrirut led her team in a call to action to address the lack of efficient testing methods that would be required to reopen our schools and economy. Her team quickly developed a fast, cost-effective, and scalable solution to increase the overall capacity of COVID-19 testing using saliva-based pool testing. This SalivaClear solution is now used by over 500 schools, community centers, shelters, and businesses across the nation for surveillance testing to quickly identify infected individuals in group settings and interrupt the chains of transmission. Her team was named a winner in the X Prize Rapid COVID-19 Testing Competition for their innovative strategy and is now scaling this approach across the country. Wellesley has been working with Mirimus for our COVID testing since October, so we're so excited to have you here tonight. Thank you so much for being here. The hope is that you would talk a little bit about what got you interested in a STEM career, walk us through the scientific rationale for COVID testing, and then let us see actually what happens to our saliva samples in the lab. I'll be monitoring the chat and the questions, and then we'll have, if there's any clarifications, I'll interrupt you briefly, and then we can have questions, more questions and answer at the end. Thank you so much.
Great, thanks. So thank you for that. So I actually prepared a little lecture for you guys, and maybe I won't go into all the details, but to kind of give you an insight into, what are the different types of testing, what we were doing in the beginning and how we got here, and, why we got here. So if that's okay with you, I will briefly, maybe over the next 15 minutes or so, give you this slides that I have prepared. And then James Lowe will kindly walk you through the lab, and we'll give you sort of a at each station, what is being done. So I think the Wellesley samples are coming in today. I don't think they're there yet, but it'll be nice for you guys to have a little glimpse into that. So just to give you a insight into, Mirimus and where we are located in Brooklyn, New York, and we're right next to SUNY Downstate Medical Center. We're located in their biotechnology incubator. And this was one of the communities really hit first in the nation, and we had just experienced so rapidly, what COVID can do to your community. And it was just something we needed to do. And so, Mirimus itself started a foundation called Mobilizing Foundation through generous funding from the Skoll Foundation to, again, set up testing and also research into, what is COVID-19, what is SARS-CoV-2, and what it was doing. So I think, unless you've been hiding out in some place, I think everybody knows what SARS-CoV-2 is, and they know what COVID-19, which is the disease caused by SARS-CoV-2 is. And, what's a little bit scary right now is that the number of cases are rising almost more than 1 million a day, just in India alone. And we are experiencing and have now experienced more than 3 million deaths across the globe. And although it seems somewhat contained here in the US due to increase in vaccinations and testing, there are places in the world that are experiencing tremendous number of cases and deaths right now. And so we really do have to keep our eye on this and not forget, that we need to stay vigilant. So we all know what has happened to schools, businesses. We've had worldwide shutdown, and it really is estimated that nearly 60 to 80% of our population in the globe will become infected. And the mortality rate is still not truly identified. But again, I think it's really skewed depending on what risk factors you have, whether you're in the elderly population or whether you have some type of pre-existing condition. So because of all of this, we know here at Mirimus, and your group knows as well, that robust testing really is key to curbing transmission rates, and particularly as new variants come on board and enter our borders, we will need to surveil for them as well. So this was in the early days of the pandemic. This was us tag teaming, really with the clinicians at the hospital and developing a blood bank. And what we had done initially was thought that we could actually test for antibodies in patients and determine, whether or not recovered patients had antibodies, whether that serum from them, which would be called convalescent plasma, could be used to treat critically ill patients. But also at this time, this was about, April and March and April, it was thought that, once someone had COVID, they would have immunity, and they would be able to have an immune passport, and we would be able to go back to work and school because of that. So we started developing antibody tests very rapidly. And antibody tests, really to detect some of these. Do you have antibodies against primarily this spike protein? So COVID or SARS-CoV-2 is a virus. And as you may know, viruses cannot replicate on their own. They do require a host. They must infect our cells and then replicate that. So these proteins here I'm showing you are some of those that we utilize to test for and detect. So whether we have antibodies towards them, which would tell that you had immunity, or whether you detect whether you have these proteins being expressed or the viral RNA being expressed, so that way you have COVID and an active infection. So the main ones we look for are the S, the N, some assays look for the E, and there's also what's called the open reading frame on the RNA. Okay. So in a brief nutshell, what happens when you get infected with SARS-CoV-2 is within days, your what's called your innate immune response. This is sort of your non-specific immune response, which are comprised of natural killer cells, macrophages, and so forth. They start to secrete cytokines. They say, your body says, oh, I've been infected with something that's foreign that shouldn't be here. And so they drive up this inflammation and by secreting cytokines. And then they will actually recruit your T cells to come over and take a look at the cells that have been infected and try to destroy them, knowing that they really don't belong there or they've been infected. And so your innate immune system starts in days, it kicks in and your T cells respond in the first week or two, and then it will recruit your B cells, which are the ones that are your memory. They will make antibodies usually within about 14 days or so, to then fight off that disease if you're ever reinfected. What we saw was really interesting in the early days of COVID. What we saw was this was not the typical response. People were having an overactive innate immune response, causing a lot of damage. The antibody response was not within 14 days. It was sometimes not happening till 20 days, 30 days, 40 days, sometimes even 50 days after someone had been infected. We could not detect antibodies. And more so, we were seeing that high levels of antibodies were only present in those that were hospitalized and had severe cases. But those that only had a mild, or they were even asymptomatic, had no symptoms, were not really developing this response. And so we realized that we would need to know, if we couldn't detect antibodies, we would need to know, do they have COVID? Are they infected? So I just talked about antibody detection. There are two other types of tests. One is an antigen test, which will tell you, do you have viral proteins? This one can be done and it's rapid, but you're not amplifying, so it's not as sensitive. And then the ones that your samples go through are this nucleic acid test, where we actually will take and isolate the viral RNA, which you'll see in the laboratory, and we'll amplify it and then determine how much virus you actually had in your sample, so we can determine whether you had a current infection. So this is just a brief in a nutshell of the different types of testing that we have. And not to go into too much details, but this looks a little bit complicated. But if you're looking for antibodies, you want to test antibodies, what you'll do is you'll make synthetic viral proteins and you'll immobilize them, as I'm showing you here on the right-hand side, to something. And then you'll take a patient's blood sample and you'll put it on top of those synthetic proteins that are the viral proteins. And if it sticks, you'll know that that person has antibodies. On the left-hand side, to detect if someone has an active infection, you'll actually isolate the RNA, have it reverse transcribed into DNA, and then copy it over and over through a process called PCR, polymerase chain reaction, and then look to see how many copies did I actually detect. And that's what we actually do in the lab, because again, we realized early on that measuring antibodies was not really a good way to determine whether someone had immunity or not. With the rapid antigen test, the difference with that is it's kind of like a antibody test, but it's the opposite. What they will do is create an antibody that actually, it's a synthetic antibody, and they will immobilize that, and then they'll take your sample, whether it's saliva or a nasal swab or anything like that, and put it onto these antibodies. And if that sticks, it will tell you that you have viral proteins there, and you can detect it. So those tests can be good. They can be done at home. They are rapid. However, because you're not actually amplifying anything, they're not as sensitive. So we have seen that these can miss almost 1 out of 3 active infections. And therefore, we rely on the PCR test more. So this PCR test that I've described already, again, just showing you in another way, is you can take a sample and you can isolate it, the RNA, and it has to be converted back into DNA because DNA is much more stable, and you can amplify it. And once it's converted back, we can then amplify it and detect, do we have the presence of multiple different viral genes or not? So this type of testing takes anywhere from at least an hour if you have a machine nearby, or it can take up to multiple days. When we get your samples in the lab, we really try to expedite that and have the samples tested on the same day that they arrive, and the results are back to your school within 24 hours. So, what are the key things that we need to build a really robust COVID-19 molecular diagnostic pipeline? Well, all the things that I've described to you guys. One It needs to be fast. We have to have really rapid turnaround time. It needs to be cheap. In the early days of this pandemic, and what really pushed Mirimus to build a pipeline this was because tests were so expensive. They were hard to get. And even in New York, we had to have symptoms, or we had to have traveled to China or somewhere, that had disease already detected. Otherwise, we couldn't get it. And they were somewhere around the realm of $150 each. But what really struck me is they needed to be easy. And what I did not like, and my children did not like, was this nasal pharyngeal swab. And that was, if my children saw one of these, they would run away. So we needed something that would work, that would be simple, and you guys would be able to do by yourself and collect on your own. And so, there are some that do nasals, these anterior nasal swabs are softer. You only need to collect sample from the front of your nose, but that still some of the younger kids can't do. But what we found was that saliva, most of you by now have this done and are very capable of doing this quickly, is to just spit into a tube. And so we started testing that. And this is, one that we've seen that most people are okay with doing repeatedly and participating in testing if it's done that way. So this has a lot of words on here, but the basic principle of what I'm trying to say to you is that we wanted an approach that would be saliva-based because it was easy to do. You could do it on your own. We wanted something that would be fast in turnaround, something with high sensitivity, such as PCR testing that I showed you, unlike the antigen testing, and something with high specificity. So this is all wrapped up into what we developed, which is that pipeline called SalivaClear, which your samples run through each week in our laboratory. So what is this method? Well, when we were looking at testing, we realized that in order to bring the cost down and to do the number of tests we do, which is now in the realm of, on average, more than 15 to 20,000 individual samples a day, we needed to actually come up with a method to do it cost efficiently. And with little machinery, how could we do all those samples? The only way we could figure out how to do that was to do what we call pooling, where we would take your samples, take a little bit from each of them and pool them together to make an individual pooled sample, a pooled sample. And we would test them in pools. So if the pool is negative, all of the ones tested in that pool would be considered negative. If it's positive, we would go back to your original samples and then test them individually and then report them out. So this is a process that you guys are probably very familiar with by now, but that's really how the pipeline flows. So it's first a pool test, and then an individual test to then report out your results. This here on this slide, what I'm trying to show you is that if we were to actually test all of you, 24 individuals individually every single week, this is how many tests we'd have to do. This is how much reagents we'd have to go through. It would take a lot of time as well. We would need a lot more personnel, a lot more equipment. But by pooling, we can reduce the amount of samples we have to test, and also the amount of reagents as well as the consumables. And so we could really turn this around quickly. And this is really important because there were days in the summertime last year where it was taking 7 to 14 days for some people to get the results back. And that was because reagents were running low. Labs were not able to get the amount of reagents and the amount of plastics and consumables they needed to do all this testing. So, coming up with a strategy of being able to conserve and preserve a lot of these consumables was really necessary. So what actually happens in the lab when your samples come in? And this is what James will give you a nice tour of in a few minutes. But just to give you a general overview before he goes and shows you all of that equipment, so he knows what he is talking about. So your samples will come individually and we'll actually have to rack them. And so each of your vials, as you will know, have 2 barcodes on them, one on the side and one on the bottom. That bottom barcode is very important to us because it allows us to actually take a snapshot of all these tubes racked. So we do not have to individually scan them. And that's really important for turnaround time. Once they're scanned into our system, they go onto another machine called a decapper. Now this decapper sits in a biosafety cabinet so that samples are only opened in this biosafety cabinet to protect our workers. So that way, if there is any positive samples in there, it's really protecting them because of the airflow can only go one direction. So their hands fit into this biosafety cabinet and they're really protected from any type of infection. Once the samples are decapped, it goes into a pooling station, which will take, again, It will mix in a solution to kind of break up the your samples, which can be somewhat viscous. And then it will take a small amount from each of the samples and then put it into a reservoir. And it will do that for up to 24 samples. That reservoir of the pool sample then gets poured into a larger tube called a pool tube. So we can then take these pool tubes and now run them as any individual sample would. The key thing here is that we can run either in this Kingfisher extractor 95 individual samples, or we can run, as we do, 95 times 24, which is more than 2,200 samples at one time. So that is really how we maximize the efficiency of our machinery. So this machine here, which James will show you in there, it is a automatic RNA extractor. So what it will do is it has a large magnet, and that magnet takes advantage of the negative charge of that RNA. And so it will wash away all the proteins and everything else in the sample and allow the RNA to be stuck to that magnet and everything washed away. And then it will release the RNA into another solution. So it moves from a plate to another plate and so forth, carrying that RNA over into a clean plate that we can then have just clean extracted RNA. What we've done next after that is we take that viral RNA and we put it on into the PCR machine. And this is called a qPCR because we're doing qualitative. We can quantify actually the amount of virus that's there. So we have these automated systems, as you'll see there, to avoid, what they'll do is pick up 95 or 96 samples all at one time to avoid any human error. And that will go into a 384 well plate here, as you see here. If you were to try to do this one by one, you would definitely make a mistake. We would put from one well to another, and it's really hard to with precision to do that manually. So we do all of this using these machines so that we can only put your sample into a specific well. And then that is recorded on our software. So all of that then goes into a qPCR machine that then will do the amplification and read what that looks like. Okay, so, all right. So this is just some of the data, and this might make no sense to you. The only key thing I want you to take out of this is that because we are amplifying, and I'll have James show you some of the plots on the results as well. But because we are amplifying with each cycle, the lower the number, the lower the cycle number, okay, the higher the viral load, right? Because that means that an early time point in the cycling, we are detecting virus. So we do a total of 40 cycles. And if we can detect it, and sometimes the lowest cycle number we can detect is usually, on average, it's usually in the teens or the 20s, but sometimes with a higher, very high viral load, we can detect it even early around 11 or 12. Okay, so what I'm showing you here on this plot is that these are the cycle threshold, right? The cycle number and for each of the genes that we detect in our assay. So in a pool, we'll normally have a higher cycle. And when we then go back to test your individual samples, that will shift by usually about 2. So we will see that shift and we will know that, you know, from a pool to an individual, that really makes sense. And that's the typical, what we see for the results, okay. So I've told you a lot about different technologies, you know, some of the newer ones that I just want to bring to your attention, which are really cool. This is called the RT-LAMP. It's also a amplification process, but instead of having to have all that machinery that we do in the lab, we can actually utilize heat, and we can stick these tubes just in a water bath. And if there is virus, it will have this proton exchange, and because of the acidic pH of the solution changes, so does the color, right? So the more virus there is, it'll turn to this yellow, and you can see over time, you can detect that. So this is something that James actually has been working on in the lab, but I think it's really cool because even in classrooms, you would be able to do this in about 30 minutes time. And so this is something we're also working on that I think is just a fun thing to know, that, you know, we are trying to accelerate how testing is done. One problem with this is we still haven't been able to get to this really low viral loads to be able to detect it very clearly. So that's something we're still working on improving. So that's it. That's our team here at Mirimus that tests your samples every week. And if you have any questions, I will take those, and James can take you on the lovely tour of the lab. I see one sample here, or sorry, one question here, which is asking which is generally more accurate, a saliva or a nose test, and why? So it's commonly thought that the nasopharyngeal swab, right, the one that goes all the way back, is the gold standard, and that has been the common thinking for the last, I don't know how many years, for respiratory viruses. I have to say right now that that thinking is changing because of the research that has been done, not only in my lab, but also at Yale and other labs using saliva. It's now shown that these anterior nasal swabs, the ones that don't go all the way back, but unless you actually have some sort of viral like symptoms, you have some nasal drippage or that, it may not pick anything up, right? And so that's one of the things we are worried about in the lab. The other thing, there was a paper recently that showed that when they were following people that were not detected, and they were following for quite some time, they actually found that they could detect virus in the sample, in the saliva samples, almost 1.5 to 4.5 days before they could actually detect it in the nasal swab. So they were doing this side by side for days, following people, and they actually showed that. So I thought that was very interesting. That study just came out of Caltech. And so I can tell you that the epidemiologist will tell you the nasopharyngeal swab, the one that goes all the way back, is the gold standard, and then the one in the front is better. And they're having somewhat of a pushback against saliva, but for SARS-CoV-2, we're really showing that saliva actually tends to have even higher levels of virus. And so we're sort of changing that paradigm right now. There was also a recent publication that showed that SARS-CoV-2 actually invades your salivary glands when you have an infection. And so that could be also why we detect high levels of virus in your saliva. So I think that we are right now pushing the paradigm into saying that, maybe nasopharyngeal swabs, particularly for this virus, might not be the gold standard. Why stop at 24 in a pool? So we can actually retool our platform. We have shown that even 36 in a pool and 48 actually do have pretty good sensitivity to detect one individual within that. And we are comfortable with that. However, if there are a lot of positive, the positivity rate is too high, then we have to go back and retest all 48. So it really depends on the positivity rate that is in the community or at the school. But I think as time goes by and we feel more confident in that, we might be able to increase the pool size. It's really has to do with the regulatory bodies. A lot of them are not keen on going beyond that. We are one of the labs doing this level of pooling, and we really haven't gotten the approvals to go further, mainly due to the concerns that we might miss someone in it. But again, we have shown that in our equipment and using all of that, we can detect even that. And we can retool our equipment to do higher sample volume. So we could actually do a 10-fold more amount of volume. So we think in the future, we might be able to do a whole school in one test, pulling hundreds of them. But that really depends on whether or not the positivity rate is decreased, and that way we can do that. Okay, another question. Have labs invested in testing equipment that won't be used when the pandemic passes? Absolutely. There's a lot of equipment that is out there now that's been put into labs, and there might be excess equipment. And so we actually had this discussion at our back to school conference that happened a couple of weeks ago. And what would we do with this? Some of the answers for, this would be great to put in educational labs, to put in research labs that, and also maybe to have distributed to other countries that are going to be in need. That is one thing we could do. Another thing we could do with the equipment is to really think about how to change diagnostic testing in the future for not just SARS-CoV-2, but for other pathogens, for other types of diseases we'd like to detect. There may be other uses out of them, and we're really thinking hard about that for the future. Okay, I'll answer a couple more questions and I'll let James go on his lab tour. So I have one question here that says, during testing, what does amplifying mean? And why is it necessary for that to do accurate results? So what amplifying means, and I'll go back to this right here, if you can see this. So what we do is, because we may only have a very, very low number of viral copies that we've extracted from your sample, we want to be able to measure that, but it would be really hard to see if we only have a few copies. So what we do is we copy it. So we make a copy of that, and then we continue to make a copy of that with each cycle. So we'll copy it and copy it and copy it. So you can see if we have one copy, it becomes 2. We have 2 copies, in the next cycle it becomes 4. We have 4 copies, then it becomes 16, and so forth. So it's an exponential amount that we're actually creating. So it makes detection a lot easier. If you only have like 2 or 3 copies within your sample, we wouldn't be able to detect it on our machinery. And that's one of the problems with the antigen tests, is that if you have very, very low virus, you may not be able to detect it. So that's why we amplify it, to make sure that we can see anything that's there. All right. So false positives, there's a question about false positives. One thing that we do with false positives, or why we have very few false positives, is because in this testing, let me see where I'm at here, right here. Okay. One of the things we have low false positive in ours is because your sample, to be called positive, has to go through 2 cycles of testing. One, it has to be found in a pool, first of all. And secondly, we test it again. And so, you know, we're pretty certain after that, that it's also that we're not going to have a false positive because of that. A second reason why we don't have a false positive is because the assay that we're using very specifically only detects SARS-CoV-2. And not any of the other viruses, because it uses a very specific primer probe set to that genetic code of the virus. And so that's why we know that it's very specific for that, and it doesn't detect any other virus. So the manufacturers of this test kit have tested it against, 40 some other different pathogens and viruses, and they know that we're not detecting something else. We are detecting only SARS-CoV-2. So someone's asking about business model. And yes, we are very committed to testing communities and helping out. And it really works because we have our foundation and we work hand in hand. So those communities that may not be able to afford testing can apply to the foundation to get support for that testing that we can pair up with. As a business and as a biotech company, we do have investors, and so we do have to make sure we're not losing money. So that is one key thing. And so that's how we can really bridge what we do for our community, as well as, building a business so that we have common goals. So that is one of the reasons why we founded the foundation from the very start, so that we could also receive donations to help this community effort. But still, as a business, we do have to make sure that we can make ends meet, that we can continue testing and, build the business so that we can continue to further, expand and provide more testing for even more communities. Okay, I'll take the last question here, which says, how does food or brushing teeth influence the results? Well, brushing teeth, you do have some components in there. Particularly mouthwash has components in there that can transiently kill the virus. So if you do that, we will not have a good sample. You might have killed the virus off before we can detect it. There's a lot of things that may, yes, inhibit that, especially in your toothpaste as well. Food, we don't know if particular food particles will actually prevent the testing, and give us some false results. But what it is, it's pretty gross. I can tell you that, to see your food in the samples. That's one thing. And 2, it will clog the pipette tips. So we do not want food particles in there, because that will really clog the pipette tips and slow down our testing process, and then they have to go in and clean the machines and so forth. So that's where James will say, yes, please do not eat and do not leave food particles in your samples. All right, thank you all. I will let James take over from now on, and I will stop sharing. And James, you can take them on this lovely tour of our lab.
All right, hello everybody. I can definitely attest to the fact that seeing food in the pipettes is very gross, so please don't do that. Another thing is citrus foods or juices, things like that also have a lower pH, which could start to affect things potentially. All right, so welcome to Mirimus's testing facility. I will start giving you a tour. So when we first enter, we actually have our intake area, and this is going to be the area where we first start processing your samples. So they'll come to us and they will be, shipped in something like a bag, much like this, and they'll come in these little sample containers. And then we'll actually have our intake team take those out and start racking them on these barcoded racks, which you can see right here. And what these are going to do is they're going to associate, and we're going to scan those into our system. And what that's going to do is it's going to associate the rack with your samples. And then it's also going to use one of these pool tubes, which is actually where we're going to combine all your samples at the end of the process. It's going to associate all those together in our system so we can keep track of you and your spit. And it's going to kind of help us throughout the process to, make sure that we get the results to the right people at the right time. So after that's all done, what's going to happen is intake is going to take the pooled samples and bring them into our testing facility. So
can you show them one of the scanners and how that works?
Oh yeah, of course. So we have kind of a multitude of scanners, but here, I'll show you this one. So this is one of our scanners. Back here is a camera, which will actually scan the barcode on the rack. And this whole screen here has a bunch of cameras at the bottom, which is going to scan all of the barcodes that are on the bottom of each tube, like that. We're going to put it into the system. Here, May, are you scanning anything, Steve? All right. So we're going to put it into the system. That looks much like this. Maybe we'll get a scan for you. Each company has its own associated code, which we use to identify them. And then we're going to put the rack on a scanner, and it will scan everything, all the barcodes on the bottom, associate it with the rack, and the file should pop up and look something like this, right? And that's going to have all of the barcodes laid out in, it'll write all the barcodes out and give them assign them a position within the rack. And this will correspond to the actual position of them in the physical rack here. We'll label that. And then once that's done, we can shuttle it into the lab, which I will show you now. So this is our actual testing facility where we do, well, whole thing's a testing facility, but this is actually where all the cooling, things like that happen. So let's take this rack, for example. This is all of the finished samples that have been scanned in, and we shuttle them in here, and they're picked up by one of our pooling teams, right? One of the goods that we actually use to do all of our pooling. And I'll show you some of the machinery in here. So this machine right here is our decapper. It goes through decapping and recapping processes. As you can imagine, there are 24 samples per rack, and we get countless racks a day. In fact, it's not uncommon for us to hit 15,000 to even 20,000 individuals a day. So decapping all of those would be brutal, and it's much easier to use this kind of machine. It saves on a lot of time, so you can see the process going on right there. So after the rack is picked up and decapped, it's passed over to this person over here who attends to the pooling station, right? So you can see all of these are the decapped racks over here. And the pooling station actually has the important job of taking all of those 24 samples and combining them together into the single pool tube that I showed you earlier. So the process of this, or the way that this works, is it actually has a multi-pipette up here, which you can see, and that's going to go across and do some of the pipetting. That's going to do some of the pipetting for us. Now we can occasionally implement a manual part, right? So we'll work alongside the machine to help make things go a little faster, which you can watch. So this is the step where it picks up DTT and it's going to place it into the actual samples. Now, if you remember from the presentation, DTT is the substance we use to make the sticky and gross saliva just a little less viscous, a little easier to work with. So you can see that it's doing multiple pipettes to make sure everything's mixed in. And then once it's done, it'll come back down, pick up the sample, and deposit it into this reservoir over here. Now, what happens after it's deposited into the reservoir is we manually mix it, and then we put it once again into the associated pool tube that you can see here. So that this pool tube is going to contain a small amount of all 24 samples that are in each rack. And then what happens after that is we're going to take this and we're going to extract all of this or all of the RNA from those samples. Now, before I go and show you the RNA extraction machine, these hoods are, I don't know if you guys remember from the presentation, but these hoods have this wall of air that's actually shot up from down here. You might be able to hear it, but it does get quite loud. But what that does is that actually, if you guys, I'm sure you all know that the primary way that COVID-19 is actually spread amongst the population is via inhalation of viral particles. And so this helps to kind of block any of those particles from getting out and protects the people who are working at each station. All right, so after we have collected, we collect the pool racks on other racks of about 48 per rack, and we use 2 of those to put into a 96 well rack, which we then shuttle over here. So this hood contains all of the plates that we're going to need for our extraction, right? So our RNA extraction, which we use this machine for. Now this is known as the Kingfisher, and what this actually does is it uses charged beads, charged magnetic beads, to actually take the it'll take the sample plate, which we have placed, or we have loaded all of your samples into, right, with the magnetic beads. It'll go through a wash cycle, an ethanol cycle, which acts again as another wash to make sure that all of the RNA is pure and clean. And then the RNA will come out in this elution plate. And so what we'll do after that is we'll take that elution plate and we'll prep it for PCR. So is there anything else I should show in this room before leaving to go show the PCR?
No, but why do we have them separated, the samples from the PCR room?
Ah, yes, that's important because samples PCR is incredibly sensitive. In fact, I think our PCR is sensitive up to 1 copy per microliter, which means it can detect 1 copy of the virus for every microliter that we have of saliva or sample, that we're testing. So with something so sensitive, we have to be very, very careful about contamination. So we actually keep the PCR room as far away from the sample room as possible, just in case there's a spill or some viral particle gets into the air and contaminates our elution plate. We want that to be as pure as possible to make sure that there are no strange results that pop up when we actually run the PCR. So this is actually perfect timing because we have an elution plate right here. So this is actually full of extracted RNA that we're going to prep to get put into the PCR and actually run. So there are a couple reagents that we use here. One of the more important ones is this nice, pretty purple reagent. This is actually the primer that we're going to use that's specific for the COVID, specific for SARS-CoV-2. And that is going to be that's the very specific primer that's going to detect only the COVID viral samples and nothing else. So yeah, we'll get the master mix ready, and we'll actually use this machine, which is my personal favorite. It's called the Mantis. Now, this is going to set up, you can see here, this is a 384 well plate. A lot of different samples can fit in here. And we're actually going to use this device to set up that plate without having to pipette manually all of these wells. Now, as you guys can imagine, pipetting all these manually would take not only a lot of time, but it would also leave you with carpal tunnel by the age of maybe 25. I'm only a couple years away from that, so I'd really prefer not to get carpal tunnel anytime soon. That's why I love this machine so much. So I'll show you guys a little demonstration because this actually needs to be cleaned. See if I can get something ready for you. So this machine is pretty much just a liquids handler, and what it's going to do is it's going to do all of my pipetting for me. so that I don't have to worry about it. Here, I will see if I can get something set up. So I don't know if you guys have ever watched those nature shows. I know it was Earth Day soon. I think Disney Plus did something, some nature documentary for Earth Day, but if you guys have ever watched any sort of like bug documentary, which I know it sounds kind of lame, but they're actually pretty interesting. You guys know what the praying mantis is. Now, the reason this is called the mantis is because it has this really interesting arm that looks and acts a little bit like the arm of a praying mantis. Now, I'll show you guys what that actually looks like running. We'll do this. So what this machine is going to do is it's going to use its little praying mantis arm to pick up our chip with all of the liquid reagent that we're going to use. And it's going to start, let's use a paper towel here, but it's going to start actually dispensing it. So it might be hard to see, but it is actually dispensing small volumes. This machine is incredibly accurate and it saves a lot of time. So this is one of the machines that we've kind of used to expedite this process, to get everything, or to get this process to a place where we can actually get results back to the customers within a 24-hour period. You know, pooling was part of that. This machine is part of that. Kind of the whole thing has been streamlined to make it as easy as possible to get you guys your results as fast as possible, so that you know what steps you can take in the after testing, right? Like if you're positive, negative, whatever it is. So that's part of the whole process that we've set up. Next, we're actually going to load that 384 well plate with the samples that we've set up, right? We use this machine, which is another, it's called the ViaFlow. It's another machine. And what this is actually going to do is it's going to come down, well, we don't have any tips in there right now, but it's going to pick up some tips, come down, and help us transfer all of our samples into a plate. So let's see. Here we go. So yeah, it's going to pick up tips much like this. And you can see there's a lot of tips here. It's going to come down into the elution plate, which we have set up right here. It's going to come down, pick up the solution, and allow us to transfer it to a 384 well plate very easily. Now, I'm not going to do that right now because there's still a couple steps that we actually need to go into that particular elution plate, but that's part of the process. So the next step of the process after that has been, after the plate has been ready or made ready with all of the master mix and all of the sample, we take it over to this machine. Now, this machine is known as the Quant Studio, and this is actually our thermal cycler, which will take care of the PCR and read our results, right? So the way that this works is, the way that thermal or PCR generally works is that it goes through a series of heating and cooling steps that kind of denature the DNA, allow the primers to anneal, and then allow polymerase reaction to occur so that you can amplify your DNA pretty quickly. The whole process in the Quant Studio takes about an hour and 10 minutes, I think, is the cycle time. And we can get the results as soon as that's done, we can get the results and we can actually read them. Now the results.
I'll share my screen right now.
Oh yeah.
And show them what it looks like. Yeah. Okay.
Oh, you got it.
Yeah.
So,
yep. And I pulled up a positive result right here for them to see.
Ah, perfect. So, so the results look a little like this, and I know it looks a little confusing at first, but it's actually not quite as difficult to read as you might expect. So you can see that there's a series of lines or a series of curves that you can see on the graph, right? I can actually, oh, there we go. So this one, or they're color coded, and this one in particular refers to the N gene, right? So the N gene is one of the targets on the COVID virus, right, that we're trying to detect. And so PCR, or if there is a positive sample, you expect to see that this N gene will actually amplify, and that's what this curve is telling you. It's telling you that there is amplification on an exponential level and a detectable level of this N gene within this sample. Now, on a normal and negative sample, you'd expect to see only two curves, which are our positive controls, right? That would be RP and MS2. However, if there's any more curves than that, it could be the N gene, the ORF gene, or the S gene. In this case, this is actually an S variant. See the S gene, a mutation of the virus, which is actually becoming more and more prevalent, unfortunately. Yeah, so if for a positive sample will actually have more than 2 curves, is the simplified way of thinking about it. They'll have an N gene and an ORF gene curve, whereas a negative sample, we shouldn't see any amplification at all. And so it's actually not too bad. So I guess, yeah, well, so this is a negative sample. And I know you see some curves here, but they actually correspond to our 2 controls, right? That's RP, which is a gene that all of you have in your regular cells. It's a housekeeping gene. So it should be there. And the other one is MS2, which is something we actually put in to make sure that the process is running correctly. So in a negative sample, 2 curves. In a positive sample, you'll see more than that. Yeah. So compared to this one, where you notice that there's a lot more than just 2 of those curves that are being amplified. So that's actually how we start to, or when we take this data and we can send it back to you, we can give the CT values. We have a whole bunch of other statistics that we can read from, and we can let you guys know whether or not your samples came back positive. All right, I think, is there anything else that I should go over? I think that's the most part of the process.
No, I think you did a great job there, James. Thank you.
Thank you. All right, well, thank you guys very much for sticking around and listening to me babble, but yeah,
Can you tell the students one reason why you went into science?
Oh, one reason that I went into science. So, ooh, that's a good one. There's actually a lot of reasons that I ended up going into science, but one of the reasons that I went into science was because I've always had this kind of, for a while I was taking a look at the systems that we have in place, and medicine, and there's a whole bunch of different systems that we have in place that I feel like, they're fantastic systems, but I always wondered if there were ways that we can make them a little better, right? For me, one of the reasons I started getting into science is actually because one of the things that I noticed was in cancer. I noticed that there was a lot of different systems that, it's a really tough disease to deal with. And there's a lot of different ways that I feel like we could improve upon it. And, it's a slow process, but I wanted to really learn about that. So I started learning about science, and I found that I really loved it. And I wanted to try and make improvements in different fields all across the spectrum. So that was one of the things that really hooked me into science. But there's a multitude of reasons.
Well, great. Thanks for sharing that with us. I think for myself, it was just, my mother was always in medicine, and I really enjoyed the creating something that could help medicine, on a larger scale than a 1-by-1 with patients. And so that's really why I got into this field. And just to maybe go a step back and give you a little bit of detail about myself, I actually did a MD PhD program. So I did a bachelor's degree at UC Berkeley in molecular biology and biochemistry. And then I went on to do some research at Mount Sinai, and then decided that I wanted to do a joint MD and PhD program. In that type of program, you will do 2 years of your medical degree. Then you will go on to get your PhD. And then after that, you'll finish your last 2 years of clinical work. I had a really interesting diversion from all of that, because after my PhD, I'd actually started this company. I had really figured out a way to expedite the genetic engineering of animal models to study disease. And so I was offered an opportunity to start this company, and I went ahead and did that. And then took a leave of absence from school. It was 4 years later that they said, go ahead, you need to finish medical school or resign from the program. So I went on and took a dual position running the company, and I went back to medical school to finish that. But after I'd finished medical school, I realized how interesting, being in the sciences in the research laboratory, that we could develop things for the clinic. And so I wanted to continue on that. So I went back to the company full time, and I've been running this company now for about 11 years. So.
Thank you so much. I really appreciated that. If there's any questions, anyone can feel free to type them in the chat. We did have a couple of questions pre-submitted, which maybe I could just ask you. In term, kind of two questions. How long is the saliva sample viable, and how do you dispose of them?
Sure. So the saliva samples are very stable, and we believe it's because that coronavirus has that shell, that outer shell that's really stable. And so actually we've left samples out, positive samples out on the bench at room temperature for more than 6 months, and we can still detect it. Now, whether or not that sample is still infectious, we don't know, but we can still actually detect the viral RNA in it. It does have some level of degradation, but we didn't see much degradation. We have actually taken saliva samples and put them in a winter cycle. So we'd freeze them, thaw them, freeze them, thaw them, and we can still detect the virus after that. And we've also put them in extreme temperatures beyond boiling, and have cycled them through extreme summer temperatures, and we still can detect the virus. So it's very, very stable, sample. And so we keep samples in the refrigerator. We don't actually freeze them at all, because we believe it's actually more stable rather than freezing and thawing them. And the second sample, or sorry, the second question was,
what do you do with all the saliva?
right. So with the negative samples, after 3 days, we dump them into a red biohazard bin, and they get incinerated by a company called Stericycle that takes them out and incinerates everything. So all of your samples are barcodes. We don't have any names attached to them, so we don't even know whose sample is whose. So that all goes out into, again, it gets incinerated by a company. With the positive samples, we keep them on a separate rack, and we may use them in the future to help test new assays that we're developing, or we may get special requests from your Department of Health or any Department of Health that we've reported to actually screen for new variants, and they will want to do some genetic analysis on the viral RNA. The samples themselves, though, because we've isolated the RNA, we will just send the RNA to them. So it doesn't comprise any of your DNA or anything in that material. It's just the viral RNA that's present in it that they may want to sequence to learn whether or not there's been some mutation in that sample.
And last question, unless anyone types anything in, can you speak to a little bit about the variants?
Yes, so sure, yeah, it's a very kind of frightening thing. So any RNA viruses in general mutate very frequently at high frequency. So anytime you give it a potential to replicate, it will have some level of selective pressure to want to be able to invade the next cell and also replicate at a faster rate, or even more. So it's very, very common for viruses like this one to have a high mutational rate, which is why we need to stop the transmission, because anytime it has the ability to replicate, it's going to try to mutate to make itself, again, more readily infectious and transmissible. What the variants have done, the new variants, is they've actually mutated a lot of portions on their spike protein. And that spike protein that I've shown you, the virus is called coronavirus because it looks like a crown and it has those spikes. Those spikes, that S protein, is what actually binds to our human receptor, which is called the ACE2 receptor. And it binds that receptor like a lock and key, and then it will get taken into the cell and it will hijack our cells to make it replicate itself. So it will try now, what it's doing is mutating so that lock and key is even firmer and even more stable, or it will try to even internalize quicker. So those mutations that are happening are ones that actually allow it to invade our cells much easier. And that's why the transmission rate of these variants are much higher.
Well, thank you so much. I thought that was fabulous. It's after months of spitting, it was interesting to see where the samples actually go. So thank you so much for your time. Thank you, Dr. Prem Premsrirut. Thank you, James. This was fabulous.
Great. Thank you all.
Thanks.
Have a wonderful night. Bye.
Thank you. Take care.